Imagine a vast library holding the instructions to build a human being. Each book in this library is a chapter of our genetic code, a blueprint that defines who we are. This is our genome, a complex tapestry woven from billions of DNA letters. These letters form the sentences and paragraphs that tell the story of our biological existence. The Human Genome Project, or HGP, was an audacious scientific endeavor. It was a quest to read and understand this intricate code, letter by letter, and to unlock the mysteries held within. It sought to read this intricate code, letter by letter, to decipher the language of life itself. The project was born in the 1980s, a time when the scientific community was beginning to grasp the potential of genetic research. Scientists began to dream of a future where the secrets of our genes would be revealed, where we could understand the very essence of what makes us human. This was a time of burgeoning technological advancement in DNA sequencing. New methods and tools were being developed, paving the way for the ambitious goals of the HGP. The potential benefits of understanding our genetic blueprint were too great to ignore. The HGP promised to revolutionize medicine, offering new ways to diagnose, treat, and even prevent diseases. The HGP promised to revolutionize medicine, transforming the way we approach healthcare. It held the key to understanding the root causes of diseases, providing insights that could lead to groundbreaking treatments and cures. Scientists envisioned a future of personalized medicine, where treatments could be tailored to an individual's genetic makeup, ensuring the best possible outcomes. Treatments could be tailored to an individual's genetic makeup, offering a level of precision and effectiveness previously unimaginable. The project also had profound implications for our understanding of human evolution. By comparing our genome to those of other species, we could trace our evolutionary journey and uncover the shared heritage of life on Earth. By comparing our genome to those of other species, we could trace our evolutionary journey, understanding how we have evolved over millions of years. The sheer scale of the HGP was daunting. The human genome contains over 3 billion base pairs, each one a piece of the puzzle that makes up our DNA. The human genome contains over 3 billion base pairs, the chemical letters that make up our DNA. Sequencing this vast amount of data would require a massive collaborative effort. These are the chemical letters that make up our DNA, the building blocks of life. Sequencing this vast amount of data would require a massive collaborative effort, bringing together scientists from around the world. Sequencing this vast amount of data would require a massive collaborative effort, a global partnership of minds and resources. It would demand the development of new technologies, innovative approaches, and unprecedented levels of cooperation. Despite the challenges, the potential benefits of the HGP were too great to ignore. The project promised to unlock the secrets of life itself, offering insights that could transform our understanding of biology and medicine. The project promised to unlock the secrets of life itself, revealing the hidden mechanisms that govern our existence. It was a challenge that the scientific community was eager to embrace, a journey into the unknown that held the promise of profound discoveries and transformative advancements. The Human Genome Project was a testament to the power of international collaboration, a monumental effort that brought together the brightest minds from across the globe. Scientists from around the globe united in a common goal: to decipher the human genetic code, a task that required unprecedented cooperation and coordination. The project was spearheaded by the U.S. Department of Energy and the National Institutes of Health, two institutions that recognize the immense potential of understanding our genetic blueprint. It involved researchers from the United States, the United Kingdom, France, Germany, Japan, and China, each contributing their unique expertise and resources. This global partnership was essential for the project's success, as it allowed for a pooling of knowledge and technology that no single country could have achieved alone. It allowed for the sharing of resources, expertise, and data, creating a collaborative environment where breakthroughs could be rapidly shared and built upon. Laboratories around the world worked in parallel, each focusing on different aspects of the genome to maximize efficiency and speed. They focused on sequencing different segments of the genome, a strategy that significantly reduced the time required to complete the project. This distributed approach accelerated the project's timeline, allowing for rapid progress and the ability to tackle challenges from multiple angles simultaneously. The HGP also benefited from a unique blend of public and private funding, which provided the financial stability needed to sustain such a long-term and resource-intensive project. Government agencies provided the initial impetus and significant financial support, recognizing the potential benefits of genomic research for public health and scientific advancement. Private companies eager to capitalize on the potential of genomic information also invested heavily in the project, bringing in additional resources and innovative technologies. This collaboration between the public and private sectors ensured that the HGP had the resources it needed to succeed, blending the strengths of both worlds to achieve a common goal. The race to decode the human genome was not without its controversies, as the involvement of private companies raised questions about the ownership and accessibility of genetic information. A private company, Celera Genomics, entered the fray, promising to sequence the genome faster and more cheaply than the publicly funded project. It aimed to sequence the genome faster and more cheaply than the publicly funded project, leveraging advanced technologies and a competitive spirit. This sparked a debate about the ethics of patenting genetic information, with many arguing that the human genome should remain a public resource accessible to all. Ultimately, both the public and private efforts contributed to the successful completion of the HGP, demonstrating that collaboration and competition can coexist to drive scientific progress. The Human Genome Project was a monumental undertaking, a scientific endeavor that aimed to map the entire human genetic code. It spanned over a decade and involved the efforts of thousands of scientists from around the globe, each contributing their expertise and knowledge. The project officially began in 1990, marking the start of an ambitious journey to decode the blueprint of human life. The goal was to complete a high-quality sequence of the entire human genome within 15 years, a task that required cutting-edge technology and unprecedented collaboration. One of the first major milestones was the completion of a genetic map of the human genome in 1994, which laid the groundwork for future sequencing efforts. This map provided a framework for sequencing efforts, guiding scientists in their quest to decode our genetic information. It identified the locations of specific genes on chromosomes, offering a detailed view of our genetic landscape. This was a crucial step towards understanding the organization of our genetic information, paving the way for future discoveries. In 1998, a private company, Celera Genomics, announced its own effort to sequence the human genome, adding a competitive edge to the race. Celera's entry into the race spurred a renewed sense of urgency for the publicly funded project, pushing both teams to accelerate their efforts. A working draft of the human genome was announced in June 2000, a significant milestone that brought the scientific community closer to its goal. This was a significant achievement, representing years of hard work and dedication from countless researchers. It marked the first time that the vast majority of the human genetic code had been deciphered, a monumental step in understanding human biology. The draft sequence was incomplete and contained gaps, highlighting the challenges that still lay ahead. Despite these gaps, it provided scientists with a valuable resource for further research, enabling new studies and discoveries. The HGP reached its official completion in April 2003, a momentous occasion that marked the end of a long and challenging journey. This marked the release of a highly accurate and refined sequence of the human genome, a testament to the dedication and perseverance of the scientific community. This landmark achievement revolutionized the fields of biology and medicine, opening up new possibilities for understanding and treating genetic diseases. It ushered in a new era of genomic research, where the possibilities for discovery and innovation are boundless. Section 4. A revolution in medicine understanding and combating diseases at their roots. The Human Genome Project has had a profound impact on medicine. By providing a complete blueprint of our genetic makeup, the HGP has revolutionized our understanding of disease. It has paved the way for new diagnostic tools, treatments, and preventive measures. One area where the HGP has had a major impact is in the field of cancer research. Scientists can now identify specific genetic mutations that contribute to the development of cancer. This knowledge is leading to the development of targeted therapies. These therapies can precisely attack cancer cells while sparing healthy tissue. The HGP has also been instrumental in advancing our understanding of genetic disorders. Scientists can now identify the genes responsible for a wide range of inherited diseases. This has led to the development of genetic tests that can predict an individual's risk of developing certain diseases. This information is crucial for early detection and intervention. The promise of personalized medicine is another exciting frontier in healthcare. The HGP has made it possible to analyze an individual's genetic makeup. This allows doctors to tailor treatments to a patient's specific needs. This approach has the potential to revolutionize healthcare. It can lead to more effective treatments and better outcomes for patients. Section 5. The Echoes of Our Past, Uncovering the Secrets of Human Evolution. The Human Genome Project has not only transformed our understanding of disease, but has also provided unprecedented insights into our evolutionary history. By comparing our genome to those of other species, scientists can trace the genetic changes that have occurred over millions of years of evolution. The HGP has revealed that humans share a surprising amount of genetic similarity with other primates. For example, we share over 98% of our DNA with chimpanzees. These similarities highlight our shared ancestry. They provide clues about the genetic changes that led to the evolution of distinct human traits. The project has also shed light on the migrations of early humans. By analyzing genetic variations in populations around the world, scientists can trace the movements of our ancestors. This information is helping to piece together the story of how humans populated the globe. The HGP has revolutionized the field of paleoanthropology. By analyzing ancient DNA extracted from fossils, scientists can gain insights into the genomes of our extinct human relatives such as Neanderthals and Denisovans. This is providing a fascinating glimpse into the genetic diversity of our past.